I was working off the presentation, wasn't I? I was showing you the presentation as I was talking about stuff, right? Okay, let me be sure I go back to that. Okay, so the two things that I want to deal with again here to finish out innate immunity before we get into adaptive immunity are complement and interferon. I probably said this several times last time, but the pictures that we're looking at here are out of a different book. But they still show the basic same thing. Complement is a series of proteins that are found in the plasma that react to the presence of cellular antigens, usually bacteria that have gotten into your system. There are two pathways through which complement can work. And what this is really describing is a cascade of reactions or interactions between these proteins. Originally, it was described as the classical pathway. And the proteins involved were named and numbered based on how they were discovered and how the story plays out. So it starts with complement protein 1 or C1 and then goes to C2, C3, C4, and then 5, 6, 7, 8, 9. The classical pathway involves antibodies, which is part of adaptive immunity, part of specific recognition. So the actual non-specific or strictly innate aspect of complement is found starting with the alternative pathway, which really just means complement that doesn't require the specific marking of an antibody. It starts with complement protein 3, or C3, which gets broken down into two pieces. In this picture, they only show one of those pieces, but it's labeled C3b, which hopefully suggests to you that there's a C3a out there somewhere also. It just doesn't have anything to do with the pathway. It plays a different role. C3b will attach to the surface of the cellular antigen, the bacterium, and in that location can recruit C5 through C9. Now in this picture, they're kind of oversimplifying what's happening. Really what happens is C3b causes C5 to break apart into two pieces, C5a and C5b, and one of those two, I can't remember which one, then recruits C6, C7, C8, and C9. And so the fragment of C5 and all of the rest up to C9 form into what's called the membrane attack complex, or MAC, which is really just a large channel that's inserted into the pathogen's cell membrane so that it'll destroy the cell. So in this representation below the first membrane attack complex picture, the sort of rod-shaped bacterium is covered with membrane attack complexes, and it's going to cause cell lysis, or the destruction of the cell. Basically, the way it does this, by opening up the cell membrane, it makes the bodily fluid where this pathogen finds itself continuous with the internal space of the cell, and those two solutions are not in equilibrium. Usually the internal compartment of the cell will be hypertonic compared to the bodily fluid where it finds itself. And so water from your bodily fluid will rush into the cell, trying to equilibrate the concentration on either side of that membrane. But that's going to cause the cell basically to swell up and burst, lysis. Now, that's not necessarily a great thing to have happen because there might be toxic molecules inside that cell, and if you lyse the cell, it can release toxins. And there is, in fact, a specific mechanism in certain bacteria that do that. They get into your system, they have toxic molecules inside of them, and so if complement causes them to be destroyed, they release that toxicity into your body, which then makes it harder for your body to fight off infection, and other individuals of that type of bacteria will be more likely to cause a successful infection. So really the alternative pathway is just starting with C3, breaking down to C3b, and then forming the membrane impact complex. The classical pathway involves C1, 2, and 4, and adds a bit on to that. The classical pathway is going to end with C3b attached to the surface of the pathogen and causing lysis, which is exactly like the alternative pathway. The difference is just what leads up to that point. In the alternative pathway, from this other book, not our book, they list a few things that have an effect and cause C3b to be produced. Don't worry about those specifics. I don't care that you know that, but it's just there are agents that will cause that change in the alternative pathway. For the classical pathway, getting that point, when a bacterium has gotten into your system and your adaptive immune system specifically recognizes it, marks it with antibodies, those antibodies will interact with complement protein 1, C1, which will then recruit C2 and C4, and those are what convert C3 into C3b. The numbering here is seems to be a little off, perhaps. That might annoy you. All it means is that C3 was discovered before C4, but in how things work, C4 comes before C3 in the cascade of events. So C2, C4 come together and cause C3 to break down. And then we jump to C5. And C5 through 9 form the membrane. So don't worry about the numbers being a little out of order here. That's just the historical order that they were discovered in. And also it's called the classical pathway because it was the first way that complement was described, and that's why it starts with C1. So basically, originally they discovered C1 and found that it, through the activity of a protein that we now call C2, causes C3 to be broken down. And they hadn't isolated out what C5 through 9 were before they discovered C4 also plays a role in activating C3. But once something gets named, changing the name is more complicated than it's worth. So they usually stick with the names, even though in a classroom setting like this, it can drive students crazy. It usually has some historical reason for it, which is not terribly important to how it works, and therefore just makes you keeping on top of names a little bit confusing. Now, that's what complement does. Essentially, its point is to make holes in cells so those cells are destroyed. There are a couple of other things that complement does. In the process of everything I just described, it will enhance phagocytosis, which means to attract phagocytes to the now marked cellular pathogen so that the phagocytes can engulf and destroy them more quickly. That process is, in a very general sense, called opsonization. Complement is not the only thing that causes opsonization, but this is one thing it does. The definition for opsonization is just enhanced phagocytosis. So when something like C3b attaches to the surface of a cellular pathogen, it will increase the ability of phagocytes to find and destroy that. That will actually help avoid the problem I mentioned about lysed cells potentially releasing toxins, because if the phagocyte can get there and engulf the cellular pathogen before it lyses, so that it lyses basically inside the phagolysosome, then it'll contain whatever that cell happens to have cooked up to cause problems. So the enhanced phagocytosis is really because C3b is attached to the surface of the cell. That'll interact with the phagocyte's cell membrane and make it easier for it to do its job. Yeah.
What produces the complement proteins?
If I remember correctly, they're made by the liver, like most plasma proteins. They just float around in your bloodstream until they're called upon. So sort of like clotting factors, similar. And then the other thing that complement can do is it can cause histamine release. Now the cells that release histamine are called mast cells. We saw them last time. They're activated and cause inflammatory response. So complement can actually cause mast cells to release histamine, which will increase inflammation. It doesn't say here, and I don't think in our book it says either, what causes that. Actually, what causes it is C3a, the fragment that gets lost in this particular picture. But when C3 is broken down and C3b is responsible for everything we see here, the other half of that protein then can cause mast cells to release histamine, which increases inflammation, and through inflammation, more phagocytes can come around and do their job. So that'll lead to getting the phagocytes to the local area, and then C3b is the one that actually causes the opsonization. In the online version of all of these presentations, which has the pictures from this book rather than the pictures I'm showing you here today, this particular text slide looks a little bit better. Back when I first made this presentation, either I didn't know how, or this Prezi presentation software didn't make it possible. And so when I wanted to make nested lists of things, I had to do it basically by hand, and that's why this is all kind of messed up. This looks much prettier because either I figured it out or it's now a feature in Prezi that I can make nested lists within a text slide. So the fact that this one's a little bit up, which drives me crazy, has been fixed. So there are two pathways within complement. This text slide basically summarizes some of the things I want you to get out of this. The classical pathway, the first pathway that is described, requires an antibody to get started, which means that it's actually piggybacking onto the specificity of adaptive immunity. It involves complement proteins 1, 2, and 4, which they're just involved in the classical pathway. It leads to membrane attack complex formation, which results in lysis of the cell and opsonization, enhanced phagocytosis, to get rid of that lysed cell. The alternative pathway is non-specific, which just means it doesn't require an antibody. And C3b binds to the pathogen surface, which is not unique to the alternative pathway, but that's kind of where it starts. It also leads to a membrane attack complex formation, which results in lysis and opsonization. So that's the big point. Really, the classical pathway, which involves 1, 2, and 4, is the one that's based on a specific recognition, and the alternative pathway doesn't involve those and is not specific. Then we come to interferons. Now again, picture from another book. And the reason I say that is just that from that other book, it's mentioning 3 different interferons. I only care to mention the first one, alpha interferon. Um, Unfortunately, the book that we're using doesn't have a picture that depicts what happens with alpha interferon exactly. So I made one. And so in the online presentation material for this, in place of this picture, I made a picture because the book doesn't have one. But let me just describe what happens. When a cell is infected with the virus, what happens is the viral agent injects its genetic code, DNA or RNA, depending on the type of virus. The presence of foreign nucleic acids in a cell will stimulate transcription of a gene in the nucleus, specifically for alpha interferon. And so the cell will translate that gene into the proteins which are alpha interferon. It's basically a hormone that then get released from that cell and target other cells in the tissue, so neighboring cells through paracrine signaling rather than full-blown endocrine signaling, to let them know that there's a virus in the neighborhood and an infected cell is sending up this signal. Interferon, alpha interferon, will tell other cells to stop making proteins. The way that viruses work, they inject their genetic information into a cell and force that cell to make new viruses from the viral genes. And viruses have a protein coat, capsule, whatever you want to call it. And if the cell isn't making proteins, it can't make the viral coat, and no new viruses will be formed. Which is what this means by saying enhanced resistance to viral infection. It shuts down the ability of the virus to replicate by exploiting the host cell, and so no new viruses will be produced, and hopefully that'll end the viral infection. Now, it's a rather extreme thing to do. Shutting down protein synthesis in a cell basically means the cell stops functioning. It would sort of be like if you were to decide that you didn't want to, okay, this is going to be kind of weird. I'm making up off the top of my head, but you didn't want to spread a virus yourself to other people, and so you decide to stop breathing so that you don't exhale that virus on those other people. It's great for the other people. You don't expose them to the virus that's ravaging your body, but since you're not breathing, that's not very good for you. Okay, a cell that shuts down protein production because of alpha interferon signals is likely going to die. Okay? There might be a chance, and I don't know if it's at all common, but I'm not an immunologist, so I'm not entirely sure the finer points of this. But it's conceivable that if the viral infections shut down, the cells that stopped producing proteins might start producing proteins again and survive in the end. If that makes you feel better, that's great. But really the cells that are usually affected by viruses are epithelial cells, and epithelial cells die off on a regular basis, and epithelial tissues are very good at replacing them. So destroying a layer of epithelium so that you don't get further viral infections is probably worth it, since your body's already going to be able to replace those cells, and things will be okay. Don't worry about the other two types of interferons that are mentioned here specifically. Just there are other types of interferons, and they're also hormone type signals that are used within the immune response. So it's just alpha interferons that I want to mention because of the effect it specifically has to limit viral infections, because viruses are very different from all of the other pathogens that we're interested in that they're not living organisms themselves. They have to exploit a host cell to replicate new viruses, and so alpha interferon is a way for us to interfere with that process right at the root, so to speak. It's a rather extreme thing to do, but it can limit the spread of a virus in your tissues. So that brings us to the adaptive immune system. Now, to describe it, let me zoom in here. There's basically two parts to adaptive immunity, what we call cell-mediated immunity and what we call antibody-mediated immunity. It's based on two types of lymphocytes, what we call T lymphocytes and what we call B lymphocytes. The T lymphocytes are responsible for cell-mediated immunity. And it's called cell-mediated immunity because it's basically T cells interacting with the cells in our body that might be infected or diseased. And then antibody-mediated immunity is based on B lymphocytes, which produce antibodies. Now, as we think about immunity and the specific recognition of things like we just saw in the classical pathway for complement, we're used to thinking about antibodies. Antibodies are limited to just B cells. T cells have their own thing, sort of like an antibody, called a T cell receptor. Calling it a T cell receptor hopefully makes you immediately think of something that's in the membrane of a T cell, which is in fact the case. Since it's like an antibody, a T cell receptor is also able to recognize specific antigens. It just does that from the surface of the T cell. Antibodies we usually think of as circulating through the bloodstream or other bodily fluids, where they interact with things directly. In explaining the cell-mediated immunity side of things, we need to deal with what's called an antigen-presenting cell, which is a little bit of a strange thing to explain, and there's never a really good place to put it into the system. So we'll get to that in a little while. There are actually two types of T cells, what are called helper T cells and cytotoxic T cells. Cytotoxic basically means toxic to cells. And those are really the ones that interact with the other cells. In the podcast, it mentions sort of the usual way that we think of the immune system that you're taught in grade school with the attack cells. And that was sort of the way they characterized it. Attack cells, for the most part, are cytotoxic T cells. Those are the cells that go out into your body and destroy cells that are infected or damaged or diseased. They can work indirectly, meaning they can control natural killer cells and macrophages. I think I might have updated that to say phagocytes and macrophage, but those innate immune system cells, we only really talked about phagocytes, but natural killer cells are part of that too, can be directed by cytotoxic T cells. There might not be enough cytotoxic T cells themselves to fight off infected cells in your body, so they can recruit these innate system non-specific cells to help out with the specific attack on something. So they all together are sort of the attack cells, but the cytotoxic T cells are really the ones that are in charge of directing that attack. So I mentioned that I have to talk about antigen-presenting cells, but before I get to explaining those, I want to talk about lymphocyte maturation, because really what we want to do here to understand adaptive immunity is to concentrate on the life cycle of the lymphocyte, sort of where they come from, how they get to be what they are, and then what they do to help keep our body in best working order. In this, I have to introduce something that has to do with antigen-presenting cells, but I want to talk about the lymphocytes first before we get to talk about the antigen-presenting cells. It's just hard to decide exactly what to put first in this discussion. So deal with me. Some things that might seem a little strange here, we'll get to fully explain in a little bit. So first off, lymphocytes are born in red bone marrow. Lymphocytes are a type of white blood cell, and all blood cells are made in red bone marrow. The hemocytoblast, the pluripotent stem cell in red bone marrow that makes the blood cells, is stimulated to make those blood cells by hormones, and the first step is that it differentiates into either a myeloid stem cell or a lymphoid stem cell.
Myeloid stem cells give rise to all other types of blood cells. Red blood cells, platelets, and white blood cells except for lymphocytes. And then lymphocytes come from the lymphoid stem cell. Hopefully you can see there's a commonality in those names. The lymphoid stem cells then give rise to the intermediate stem cells that give rise to lymphocytes. They're probably called lymphoblasts or something like that. Eventually they give rise to either B cells or T cells, B lymphocytes or T lymphocytes. Those cells then have to mature, and where they mature differs on the type of cell. The T cells are called T cells because they mature in the thymus. That's what the T in T cell stands for. B cells stay in the bone marrow where they mature. Now, the B in B cell actually stands for something else, but for the sake of this course, associate the B in B cell with bone marrow. What the B actually stands for, if you're really interested in it, feel free to go look it up on Google somewhere. But it stands for the name of an organ that humans don't have. So I'm not going to talk about it because it's not relevant to this class. It's not an organ that we need to know. But it's analogous to red bone marrow in humans. So the fact that we're calling the B cell the one that stays in the bone marrow satisfies things okay. And I'm calling it bone marrow here instead of red bone marrow to emphasize the B, but it is red bone marrow where it's doing its job. Now, maturation, in whichever of these locations it takes place, is making sure the cells are prepared to do what they need to do when they go out to be the attack cells, so to speak, in your immune system. There are two phases of maturation. The first one's called positive selection. This next one's called negative selection. And it's selecting for cells to survive. Positive selection is testing the cells, and the ones that pass the test survive, and the ones that fail the test are destroyed. Negative selection is testing the cells to see if they do something. If they do that thing, they're destroyed. And if they don't do that thing, then they get to survive. But that'll be clear when we get to it. Positive selection only applies to T cells. B cells it doesn't apply to because T cells interact with these antigen-presenting cells, which I'm going to explain in a little while. Antigen-presenting cells present antigens with a structure called MHC. On the surface of the antigen-presenting cell is MHC attached to an antigen. And again, I'm going to get to explain this in a little while to see what it is. At this point, all I'm saying is the T cells are tested to see if they can interact with MHC. They have to be able to do that to be functional T cells. So if they do that successfully, they get to survive. And if they can't recognize MHC, then they can't do their job. There's no reason to keep them around. And so they don't survive. In positive selection, the ones that successfully interact with MHC receive a survival factor, which is essentially a hormone that's called something growth factor probably. And it allows the cell to survive. Without that growth factor, without that survival factor, the cell dies. We'll talk about cell death in a little bit after we talk about negative selection. Now, in some sources that are talking about positive selection, I'll go a little bit deeper into it, which is there's sort of three levels of reaction. T cells can recognize MHC, but bind too strongly to it, which is a bad thing. And they can recognize MHC and bind to it appropriately, or they cannot recognize MHC at all. It might, no, it's a different book that has some of that detail in it, but you might come across sources that say that. What we're looking for is appropriate recognition of MHC. So T cells that bind too strongly to it or don't bind to it at all, don't survive, because neither of them are going to be able to do their job very well. Then negative selection, which applies to T cells and B cells, is where those cells are tested to see if they recognize things that are supposed to be in our body. Now, we're trying to get lymphocytes ready to fight off foreign invaders, pathogens or other antigens that get into our body that we don't want in our body. We want the lymphocytes to be able to recognize. And the way they do that is just by molecular binding to other molecules. The three-dimensional shape of the protein that is an antibody or a T cell receptor is, let me start that sentence over again. The three-dimensional shape of the protein that is an antibody or a T cell receptor will recognize the three-dimensional shape of an antigen, which is part of a pathogen or whatever else might be getting into your body. It's sort of like a puzzle piece recognition. Antibodies are essentially shaped like this, very schematically speaking. And these lines represent protein strands, polypeptide strands. At the end of what's kind of the Y-shaped structure here, sort of where my hand is, this part here is the antigen binding site. This was kind of presented in the blood chapter where we first were introduced to antibodies. Now, what I'm suggesting here, that circle was just highlighting that area, is that the shape of this is specific to what it can recognize. So let's think about this a little bit closer up. So these are the two pieces that I'm drawing here. And I'm completely sort of making this up schematically to represent it. This one might have a shape like that, and this one might have a shape like that. Okay. Which means that it will specifically interact with something that fits into this like a puzzle piece. Now, I'm not a very good artist, so what I'm about to draw will probably not quite really match this, but let's pretend I need more than that. If I can do this on a computer, I just copy and paste things and it'll look beautiful. So, something shaped like that will exactly fit in there. Puzzle piece. The other side looks exactly the same. Okay. There are two identical places on an antigen that recognize a particular thing that's binding to. Sometimes what these are shaped to recognize are things that are supposed to be in our body, what we call self-antigens. Now, we actually already kind of met this concept before because, you have a question?
Yes. Since there's two binding sites, will it bind to antigens?
Right. Well, we'll see later on when we talk about antibodies, which will probably be on Thursday, is how antibodies can actually link two antigens together, which can be helpful.
But will it still be active if it just binds just the one antigen? Like
It can be. So like the picture of the classical pathway, the Y-shaped thing, both parts were bound to the surface of that one thing, and that attracted a complement to that thing. The complement destroyed it. We'll talk about how antibodies interact with their things in more detail later on. But it's a good thought process. Keep that in mind because it does play a part here. So back when we were talking about blood, which is actually where we were first thinking about antibodies, one of the things we learned about antibodies is their relationship to blood type. Okay. Somebody who has type A blood, on the surface of their red blood cells is an antigen that's produced by that particular gene. If this is the shape of the A antigen that's on the surface of red blood cells, and somebody makes the antibody against that, that sets up the possibility that they will recognize their own blood type as a foreign invader. Okay, so what negative selection is about is exposing T cells and B cells to self-antigens, if they recognize a self-antigen, they're destroyed. Okay, so negative selection, we get rid of the ones that do what they're being tested on. We get rid of antibodies or lymphocytes that make antibodies that recognize self-antigens. So the point of that is to keep us from having autoimmune reactions. We don't want to have lymphocytes that are capable of recognizing self-antigens so that our immune system won't attack our own body. It's not a perfect system, but it is part of the process that goes on here. Okay. T cell receptors are structurally kind of like antibodies in that they have this antigen binding site at their end. There's only one of them on a T cell receptor, not two of them, but any T cell receptor that recognizes a self-antigen will also be destroyed. Okay. And destroying that cell is a process called apoptosis. Now, apoptosis is not just this particular process. It's a very common process. And the picture that I have down here at the bottom is from the National Library of Medicine, part of the National Institutes of Health. So it's a computer-generated model of what apoptosis would look like. The word apoptosis, as its prefix is apo, which essentially means apart, and tosis. The P in tosis is silent. Tosis by itself is used medically to describe things, and some of my returning AMP1 students might, oh, never mind. I didn't do that last year. Tosis is used to describe certain situations where organs or other structures are drooping or falling. So ptosis of the eyelid is a drooping eyelid. So ptosis basically means to droop or to fall. And so apoptosis literally translates to falling apart. And so what the cell is doing is it's falling apart, it's breaking up into little pieces. The nucleus and other organelles inside the cell break down and get packaged into little spherical inclusions inside cell membrane. Okay. So the cell just completely falls apart. And then a white blood cell, phagocyte of some sort, can come along and ingest all of those little membrane-bound packets of dead cell and get rid of it. Okay. Break down the cell membrane, break down whatever proteins and whatever's inside, and recycle all those macromolecules to be reused later on. This is sort of the honorable way to die. Okay? It's called cell suicide. Cells are killing themselves in apoptosis. And they're packaging themselves up so they can be easily cleared away. Apoptosis is a common process. It happens in a lot of different situations, not just here talking about the immune system, but it's really the only place that we talk about it in this course. It happens in a lot of developmental processes. For example, you have 5 individual digits because of apoptosis. In the fetus, originally the In the fetus, originally the limb bud was a flipper. Okay. And for organisms that need flippers to swim, that's a very useful structure. But for organisms that need individual digits to manipulate for grabbing or walking or whatever it might be, flippers aren't that great of a thing. So in organisms that developed to have a different body plan, body type structures to live on land, the flipper structure that originally develops in the fetus goes away because the tissue in between the digits dies off through apoptosis. If that doesn't happen properly, then you have people that are born with webbed feet, webbed toes or webbed fingers or something like that, because the tissue might not die off properly. There was a lab down the hall from the lab that I was working in when I was a graduate student that was studying apoptosis in the nervous system. In the nervous system during development, there are a lot of neurons present that you don't have when you're an adult. Okay? There are a lot of connections that are made early on, and then through your experience as a newborn, your brain sort of weeds out the neurons that it doesn't need. And that makes for very specific connections in a functional nervous system. And the cells that aren't going to persist, the ones that made the incorrect connections during development, will die through apoptosis also. It's a very common process that takes place in a lot of development. And actually throughout your whole life, when cells need to die, they go through this. The alternative, the other way that cells can die, essentially, is lysis, what we talked about earlier with killing cellular pathogens. The cell blows up. Now, that can be a problem if the cell that's dying is at all diseased, because whatever's inside of that cell that's the cause for its disease, if that's released when the cell blows up in lysis, that will then potentially expose your body to that disease-causing agent. So this apoptotic cell might be one that was infected with a virus, and a cytotoxic T cell can come along and say, hey, you're infected, die. And it dies like this, so it can be cleared away. And all of the viruses that it was producing are trapped within those little things. They're going to get gobbled up and cleared away by phagocytes. Okay. So it's the clean way to get rid of things. Now, there's actually kind of a 50-50 split in how people pronounce this word. I pronounce it apoptosis, mainly because that's the correct way to pronounce it. But about half the people out there that try to pronounce this word will pronounce the second P, and they'll call it apoptosis. It's like, fingernails on chalkboards to me. That second P really should be silent. However, even in the lab that was studying this down the hall from me, there were people that called it apoptosis. I can't even say it. Because they just belong to a group of people that thought that in this situation, the P should be heard. But really, the root of the word is ptosis, and the P in ptosis is silent. However, pronouncing it apoptosis does suggest a false etymology, which is you could interpret what the word means incorrectly, but usefully, by thinking about that alternate pronunciation. Apoptosis sounds like the word is apoptosis. Okay, 3 parts rather than the 2 that actually makes it up. In a word that has a as a prefix, that a is usually a negative. Okay. And then pop, onomatopoeic word for things that go poop, boom, whatever. So apoptosis could suggest that the word means cells that don't go like that, okay? Which that sound would be lysis, okay? So if that helps you remember that apoptosis is not lysis, that's fine. You're not being graded on your ability to pronounce this word. So if you pronounce it apoptosis, that's okay. I'd prefer it if you didn't do it in front of me, but I'm not going to take points off of anything for you pronouncing it that way. That's what this text side is getting at, really for both positive and negative selection. The cells that do not pass positive selection, that do not get this survival factor, will then go through apoptosis and be destroyed. And then the ones that bind to self-antigens will go through apoptosis and die. Yeah.
The cells that go through apoptosis.
You had to do it, Tim.
Apoptosis. The ingredients or their parts are repurposed, or are they repurposed?
So it's, red blood cells, when they live out their 120-day life cycle, they get gobbled up. And their parts are all they're broken down to amino acids and membrane lipids, and then all those are recycled into new cells. So yeah, any of
The
this.
same thing happened with lysis.
If lysis can be contained by phagocytes, they can take care of that. But if lysis just happens somewhere and dead tissue is accumulating, that can actually be sort of food for other pathogens or something like that. So the pathogens can take it, recycle it, make more pathogens, and then things get out of control. And so necrotic tissue can actually be a site of gangrene or something like that can set in because pathogens can basically live off of the dying cells through lysis. Apoptosis, because the white blood cells can clean up after it easily, just keeps everything neater, and then those white blood cells will be able to pass those recycled materials on to wherever they need to be used to make new cells later on. Okay. Having introduced how the lymphocytes mature, now I want to go back and explain what antigen-presenting cells are, which will pick up with what MHC was. That's the thing that positive selection is testing T cells to see if they can appropriately interact with. Now, antigen-presenting cells are cells that present antigens. That probably isn't a big leap for you to understand. But when a pathogen gets into our system, we can call that pathogen an antigen because it can generate an antibody response. We can call parts of that pathogen antigens if it's releasing toxins into our system or something like that. Those would also be considered antigens. If we get something into our system that's not necessarily pathogenic, it can still be an antigen. So thanks to yesterday's storm, this isn't going to happen anytime terribly soon, but we're sort of probably starting into allergy season during the spring weather we just had. And that's because pollen and those sorts of things are being produced by plants, and now that everything is coated in snow, that's not likely to happen again for maybe another week. But pollen or whatever can get into our system, those are antigens also. Anything that can interact with an antibody, and really an antigen can also be thought of as interacting with T cell receptors. It's just kind of hard to call something a T cell receptorgen. There are two types of antigens. There are exogenous antigens and endogenous antigens. Exogenous means produced outside of your body. Endogenous means produced within your body. Exogenous antigens would be pathogens that get into your body, or pollen or something like that gets in. Endogenous antigens are things that your body makes itself. Most endogenous antigens are self-antigens, and we don't really care about those because we've already tested our lymphocytes against recognizing self-antigens and that negative selection thing. But sometimes cells can make endogenous antigens that are not self-antigens, things that aren't supposed to be there. In cancer, cells become cancerous because they start activating genes that aren't supposed to be activated, some of which cause uncontrolled growth of that cell. But there are certain cells that get turned on in cancerous cells that make proteins that cells normally don't make. In colon cancer, for example, there's a marker that can be used to tell what's going on with colon cancer for a protein or antigen called CEA, which stands for carcinoembryonic antigen. What does carcino mean? Cancer, and I'm talking about cancer. What does embryonic refer to? The embryo. Okay. And then antigen is the thing there. What carcinoembryonic antigen is, it's a protein that's made in one of two situations, either by a cancerous cell or in the embryo. Now, The maturation I just talked about is taking place in an infant, okay? Newborn to toddler age, somewhere in there. And so embryonic antigens should no longer be present. Okay? But in cancerous cells, they start activating these genes by mistake, and so in colon cancer, there's this one that's made. It starts making an embryonic protein that cells normally shouldn't make, and it becomes basically a marker for cancer. Okay, that would be an example of an endogenous antigen. Now, I said exogenous antigen, endogenous antigen, but on the screen right now, you can see exogenous presentation and endogenous presentation, because we're talking about the cells that present those antigens, not necessarily the cells that make those antigens. Exogenous presentation is cells that present antigens that are made outside of your body. And endogenous presentation is presentation of antigens made inside your body. Any antigen made outside of your body isn't going to be presented by whatever made that antigen. It's going to be presented by phagocytic cells. So phagocytes will recognize foreign invaders, engulf them, break them down into little pieces, which we looked at the end of our last class meeting. The picture I should have shown you there is smaller here. I showed you this picture before last time, right? I didn't show you this? Okay, I should have shown you this. Okay, the fact that you're saying you haven't seen this really bothers me.
Showed a different one, but it wasn't that.
Yeah, something like that. But wasn't.
So I showed you this picture.
No.
What the heck did I show you last time?
Like, I drew it. But it wasn't that picture.
Oh, I know. Okay. Okay, yeah, the one that I showed you is the one that's in the current presentation. I probably wasn't showing you this particular presentation, but it's still the same basic thing. Come on. All right, those of you watching at home, I'm sorry that you're now motion sick. Okay, so yeah, the picture I showed you last time isn't exactly this, but the same basic thing. This has got a lot more label in it, but something is pulled into the cell. It combines with a lysosome that contains digestive enzymes and acids, and it's broken down. In the picture I showed you, which actually is from Wikipedia, instead of having a container with the digestive material, it showed that it was being sort of pooped out the back of the cell. Okay. However it is, I mean, in this picture, that suggestion's kind of there. This is just showing you the remnants of what was digested. And then the picture I showed you is being released from the cell. Back to what Ollie was asking about for the apoptosis thing. If this isn't a microbe, but part of a apoptotic cell, and it goes through all of this, then what's down here at the end is stuff that can be used by other tissues, so it can be released and recycled in making other cells. But since this is about a microbe, this is called indigestible material. It's parts of this cell that can't be recycled, and it's trash. So that's what's presented on the surface of those cells. So these pictures over here that I'll get to in a second show you how presentation on the surface of the cell takes place, but that's what happens. They're shown to or presented to the T cells that are called helper T cells. The subscript H next to the T here on the next slide means helper T cell, and we'll meet helper T cells in more detail later on. But that's exogenous presentation. Phagocytic cells break down antigens that have gotten into our body from outside and presents pieces of them on their surface. Endogenous presentation is when cells in your body present their own antigens. The vast majority of endogenous presentation is just self-antigen presentation, which doesn't have any real effect, ideally because of the negative selection that I mentioned earlier. You should not have lymphocytes that recognize self-antigens. So normal body cells that are not infected with anything are just going to present signs of their healthiness on their surface. A fibroblast that makes collagen for connective tissues will show bits and pieces of collagen on its surface, basically saying, hey, look at me, I'm doing what I'm supposed to, leave me alone. Or neurons will present bits and pieces of acetylcholine receptors or voltage-gated sodium channels or any of the kind of things that we've met. Those are going to be things that are on the surface of a cell or in the extracellular matrix anyways. A cell's not going to present on its surface necessarily endogenous antigens that aren't normally exposed to the outside world. Like a red blood cell is not going to show a piece of hemoglobin outside on the surface of the cell because that's not something that should be leaving the red blood cell. That's not considered a self-antigen. If you have hemoglobin floating around loose in your system, that's a bad thing. Okay? So you don't want to ignore that. So that kind of thing is potentially problematic. But normal functioning cells will present on their surface little bits and pieces of things that they make anyways as a sign of their healthy, normal state. But occasionally a cell goes bad. We've already seen a case in this semester where a neuron can suddenly start making a protein incorrectly. The alpha-synuclein that the cells that are going to die in PAF make, that would not be a self-antigen. That would be a sign of a diseased cell. So if they could present that on their surface, the cell surface, it'd basically be a sign to the immune system that that cell's not functioning properly and should be destroyed. Okay. Or cancerous cells will present things on their surface that are evidence that they're not doing things properly, like that carcinoembryonic antigen I mentioned earlier on colorectal cells. Also, and this is sometimes a little bit confusing, endogenous presentation includes viral particles. When a virus gets into your body from outside, it's an exogenous antigen, and it can be gobbled up by a phagocyte. But if it gets into your system and it infects a body cell, that body cell is going to make new viruses. But those new viruses that your body cell is making are endogenous antigens because they're made by a cell in your body. And so the cell making the viral particles will present them on its surface as a sign that it's been infected. Okay, so that's endogenous presentation. Basically, exogenous is anything presented by a phagocyte, and endogenous is anything presented by one of your body cells. Usually it's an infected or diseased cell, cancerous viral infections, something like that. Phagocytes presenting exogenous antigens will present on their surface using what's called class 2 MHC. And then normal body cells, or body cells, diseased and infected cells, will present antigens on their surface using class 1 MHC. The type of MHC, class 1 or class 2, is important because that helps the receiving cell, the T cell, know what kind of antigen it's being presented with. Helper T cells only interact with class 2 MHC. Cytotoxic T cells, subscript C here on the text slide, only interact with class 1 MHC. So these pictures, again, from another book, are showing sort of the pathway for endogenous and exogenous presentation. So here at the bottom is a picture showing a phagocyte ingesting a pathogen of some sort, and then the lysosome merging with that and breaking down its material down to that indigestible residual material. from the little picture I showed you a second ago. Then that merges with a little vesicle that contains class 2 MHC, and class 2 MHC presents bits and pieces of the broken down pathogen onto the surface of the cell. In normal body cells, proteins that are going to be excreted or on the cell surface, like receptor proteins or channel proteins or pump proteins or collagen or something like that that's secreted, are going to be made in the endoplasmic reticulum, rough ER, and processed through the Golgi apparatus. And so part of what those secretory mechanisms do is they take bits and pieces of antigens and attach them to MHC class 1 or class 1 MHC to present on the surface of the cell. In a normal, healthy, functioning cell, that's just going to be presenting little pieces of self-antigens, which should be ignored by your immune system. Here, they show an infected cell, either it could be a viral infection or a cellular pathogen like a bacterium that's gotten inside the cell. What they don't show, you could also think of cancerous cells or other kind of diseased or damaged cells doing this. Whatever it is, antigens that this cell is producing in terms of its being infected or damaged or diseased will then be presented on its surface, which will not be self-antigens, and therefore evidence that this body cell is diseased or infected or something like that. So these two different shaped MHC molecules, class 2 here on the phagocyte, and class 1 here on the body cell, are there to prepare to present to the T cells. Which then brings us to lymphocyte activation. Now, all of the little panels here are showing how lymphocytes are activated. Lymphocytes come in two major types, T cells and B cells. T cells come in two types, helper T cells and cytotoxic T cells. So the first few panels are showing us T cell activation, starting with helper T cell and then cytotoxic T cell. And then the last panel is showing B cell activation. So depending on the situation, they can all kind of be activated in their own ways. Now, the lymphocyte maturation, the testing that I showed you before, is taking place in primary lymphatic organs, or lymphoid organs, however you want to call them, which means in the bone marrow or in the thymus. Once they've passed through the positive and negative selection, if both are required, then the lymphocytes leave the primary lymphoid structures and go to secondary lymphoid structures, which would be lymph nodes, tonsils, mucosa-associated lymphatic tissue, the spleen, et cetera, et cetera. They wait in that secondary location until they're exposed to the antigen that they are prepared for. The T cells make T cell receptors, the B cells make antibodies, but they are specific to particular antigens. Since they've gone through the maturation process, they're not going to be specific for self-antigens. They're going to be specific for things, other types of things. We might not ever be exposed to whatever the antibody is prepared for. Okay? So if you're a type A blood, let me say that again. If you have type A blood, then the lymphocytes that make antibodies against the type A antigen have been destroyed, because that would be a self-antigen. But lymphocytes that make antibodies against type B blood are not destroyed because you don't have type B blood. You might not ever be exposed to type B blood. If you get a type B blood transfusion, then you'd be exposed to it, but that's very, very rare for that to actually happen. The likelihood of you just accidentally getting somebody else's blood into your body, very, very rare situation. But the lymphocytes that make the antibodies against the B antigen, for blood type A antigen, are waiting in secondary lymphoid structures for exposure to that, whatever it might be. So once that exposure happens, then these processes are going to take place. Now, these processes are a little involved, which means they need much more than 5 minutes to explain. So we'll call it a day there, and we'll come back and see how T cells and B cells get activated in that secondary tissue next time. And on Thursday, we'll finish off the immune system stuff. We may or may not start the respiratory stuff after that on Thursday.
Yeah.